Okay, so transcriptions actually is the first steps of the gene expression. Means any gene expression, you can run away from the transcriptions. Okay, so in which particular segments of DNA, when you say particular segments of DNA, we're referring to gene. Okay, it's copied into what we call the mRNA. So enzyme involved here is RNA polymerase. Okay, so now the problem here is how this DNA RNA polymerase know where to bind and where to start. So they only choose one of the DNA strands. We do know that the DNA consists of double-stranded, so only one strand will act as a transcriptions or template strands. Okay, so if you look at this part here, transcription process, then you'll realize that the process only involves, you can see that we have the template strands versus non-template strand. Only template strands will be used to transcribe, therefore form the mRNA. So each 3-digit code here is then for the codon. Codon 1, codon 2, codon 3, and so on. Okay, so now let us look at what actually happened here, why we the RNA polymerase know where to choose and how the transcription process actually started. Okay, just a quick repeat and revisions on this. Okay, we do know that our DNA is double helix. Double helix. So particular segments of the DNA, we have the gene. Okay, the red color. So this is a gene. So if I isolate out the gene and have a look, so this gene actually, okay, start with 3' to 5', for example, then 3' to 5' here, the opposite strand, okay, anti-parallel property. So how the, oops, sorry. Wait, guys. Okay, so now those strands, so which strands would we choose? Which strands? Okay, will act as template strand. So this question actually is very easy to actually know, where if the template strand, if it is a template strand, then they must have promoter. Okay, they must have this called promoter. So what actually happened here is, now always remember ATCG not just an alphabet. They actually is a purine and pyrimidine. So they create a shape. Okay, so now look at this. What does it mean I create a shape? If let's say I have A, okay, I have T. T is a pyrimidine, right? C. So C is also pyrimidine. Okay, G. G is a purine. Can you see that the A, T, C, G created a shape? Can you see that? It created a shape here, the ATCG. If I remove, I change ACTG, AGTC, the shape will be different. So based on ATCG, actually created a shape. So this shape actually can be recognized by certain protein. Proteins or enzyme. Always remember, okay? This structure, this shape can be recognized by the protein or enzyme. So similar to here. So we do have in this 3' to 5', 5', 3' end, we do have one strand, okay? If you want to know which strand is the template strand, we find out where is the promoter. So promoter is the sequence. It consists of sequence of nucleotides that can be also recognized by RNA polymerase. Are you clear? So in this case, only one strand they have promoter. So let's say ATCG is a promoter. Let's say that, okay? So it means that if I enlarge this part, you can see that at the 3' end here, you have this ATCG, which act as a promoter. For example, I simply make up this ATCG, and not necessarily must be ATCG, okay? 5'. So means that what actually happened, the second strand, without the promoter, 5', 3'. So this strand is act as the template strand. Clear? Act as a template strand. This one we call as a non-template, just like useless one. Are you clear? Means that they don't have any function, it's just to be there. It's something like junk DNA. Are you clear? So what actually happened now? ATCG, I see that you have the shape, right? So it's recognized by this enzyme. So this enzyme is termed as RNA polymerase. So what RNA polymerase do? So after they found, okay, after they have found this promoter, what they do? They start to carry out transcription. So they form the mRNA now. So they form the mRNA. So transcription done. Okay. So what are the events? A series of events actually take place during the transcription process take place. So first step involving transcription. So we do have the RNA polymerase in general. RNA polymerase binds to promoter and initiate the transcription process. Okay, so this is general. First we need unwind. Okay, so unwind, we'll put it aside. So first, RNA polymerase then bind to promoter and initiate transcription because they can recognize a promoter. Okay, number two, so RNA polymerase can do. So RNA polymerase adds. So what they add? They add, okay, complementary free activated. Now, because we form mRNA, right? So it must be RNA nucleotide. In the directions of in the directions of 5' to 3', forming the mRNA. Okay, so how this RNA polymerase know where to stop? So RNA polymerase know where to stop because at the DNA there, we have another sequence here. Okay, I remove this mRNA. This mRNA. So how the process no stop? Because at the some sequence at the back, let's say for example, GGCCT, for example. So this GGCCT will act as the termination sequence. Are you clear? So this process will continue. Transcription stop. Okay, when RNA polymerase reaches the terminator sequence. Okay, so stop recording while discuss now. So the entire story starts when we need to do a transcriptions. We need to create this what we call a transcriptions bubble. So this is termed a transcription bubble. So this transcription bubble double-stranded. So one strand will act as a non-template strand, another strand will be act as a template strand. So how we know which strand is a template strand? Template strand must have promoter. Okay, but fun thing here is the concept of promoter is not in AS syllabus. Yeah. Right? The concept of promoter is not in AS syllabus, but I do like you guys to know about this because in A2, we keep on talking about the promoter. So either you learn it now or you learn it later. But most of the time, students will ask, hey, we have the template strand, we have non-template strand, how the RNA polymerase know which strands to be chosen, right? To be chosen, right? So in this case, very simple, that strand must have the promoter. So write down, guys, what is meant by the promoter. Promoter actually is the binding site. They are the sequence of DNA. They act as a binding site for the attachments of RNA polymerase, or the binding of the RNA polymerase. Okay, so in the exam, very important when you write the polymerase, always remember to write in either RNA or DNA. Okay, so because now we want to form the mRNA, so that's why we use the RNA polymerase. Okay, so now once you generate this bubble already, this RNA polymerase actually going to search for the promoter, bind to the promoter, and initiate. So you can see that the formations always start from 5' all the way to the 3'. Can you see that? All the way to 3'. Okay, and because mRNA is read, okay, mRNA will read it from the 5' to 3'. So therefore, the DNA, okay, you see that the DNA, we're going to read it from the 3' to 5'. Okay, so 3' here, can you see that? The strand. So go this way, transcription, can you see that? This way. So when transcription go this way, is 3' to 5' when you reach the molecules. Okay, so read the DNA molecules. Okay, so we can see that we have to break down the hydrogen bond to free the mRNA. Okay, so let's look at the answer. So first answer is T, where this RNA polymerase. Now here we have a bit of a confusion. Normally student will find out. Now transcriptions factor. This one actually is the extra. Okay, extra protein that regulate. Okay, regulates. Okay, proteins. Transcription. To regulate extra proteins that regulates the transcription. Sorry. Now, what I mean here is, okay, it depends. Now, if prokaryotic cells, very simple. Prokaryotic cells, very simple. Why? Because you can see that, okay, they just need to have RNA polymerase. Why? Because relatively, they have a very simple genome. Not complicated. So therefore, they only require RNA polymerase. But eukaryotic cells, if eukaryotic cells to get this thing done, they need RNA polymerase together with a transcription factor. Okay, so because why we need to tightly regulate our gene expression. Okay, now before I continue with this the analogy, okay, I think it's better for me to off the record. Okay, so what actually happened next? So once it bind together with one or more general transcription factor, they bind to the promoter. So it means that they form a complex. Okay, this complex tightly regulates the gene expression. It also acts as a switch, in this case, to switch on or switch off. You try to imagine RNA polymerase say that I want to have the gene expression, but then another protein say that I'm not going to support you. So therefore, it acts as a switch. Are you clear? Okay. So once you have done the first process, okay, then we're going to have the formations of the transcription bubble. At the same time, RNA polymerase now create a transcription bubble which separate the two strands of DNA helix. This is done by breaking hydrogen bonds between complementary base pair. Okay, either say complementary DNA nucleotide or base pair. Okay, so once this process have done, then RNA polymerase start to so it means that RNA polymerase start to add. Now highlight, guys, free activated RNA nucleotide. I always say that very important. There must be a free versions and they must be activated. So what's mean activated? Still remember activated? Activated must have 3 phosphate group. So hydrolysis of 2 phosphate group going to release the energy enough for them to overcome the activation energy and form the phosphodiester bond. So therefore, there is a mandatory 5' to 3' directions. Okay. Okay, so after R, then we have U, okay, U. So mRNA sugar phosphate backbone are formed. Okay, why? Because formations of phosphodiester bond. Okay, because of formation of the phosphodiester bond. So that's why we thought sugar phosphate backbone are formed, and then P. So now, when you form this, we want to free the newly synthesized mRNA strands. So you have to break the hydrogen bonds again between the RNA DNA helix. Okay. Then V. So the mRNA now exits the cytoplasm through the nuclear pore. Now, this one, it depends. If let's say prokaryote, okay, this not cannot be prokaryote, it's eukaryote. Okay. Oh, answer is wrong here, guys. It's Q and V. So Q. So in eukaryotic cells, the mRNA may be further processed. This may include the polyadenylations, capping and splicing. Okay. So this may include the polyadenylations, capping and splicing process. So this one actually involved in what we call the post-transcriptional modification. Okay, post-transcriptional modification. Okay, so after this, then the mRNA exits the cytoplasm through the nuclear pore. So answer is TSRU-P2V. Okay. So in eukaryotic transcriptions, mRNA synthesis require RNA polymerase. So don't worry about this. Okay, polymerase 2 or polymerase 3 and so on, doesn't matter. Okay, RNA polymerase carry out the synthesis in the 5' and 3' direction. This is very important highlight, guys. Always remember, any synthesis must be 5' to 3'. Okay, so it begins at the 5' end. Okay, and synthesize and then continue to add the free activated the RNA nucleotides at the 3' until molecule is complete. So therefore, mRNA strands are antiparallel. What does this mean? So if you look at this, if this is the DNA, 5' to 3', so the synthesis again will be like this. So this will be 5' to 3', okay, for mRNA. So again, it is what we call the mandatory, okay, for the synthesis must be 5' to 3'. So because mRNA is synthesized 5' to 3', therefore highlight DNA, we talk about this, right? DNA templates is read 3' to 5' direction. This is very important. Are you clear? If the question gives you 3' to 5' or 5' to 3', then you have to understand that if DNA, it must be read 3' to 5' direction. Unless the question is very nice to you guys, didn't put in any directions, then always left to right. Okay? So in both bacterial and eukaryotic cells, the nucleotide sequence in DNA to which RNA polymerase initially bind known as promoter. Okay, so means that your RNA polymerase bind to the promoter. In eukaryotic cells, okay, so most of the times you need to have the presence of transcription factor. So this is in A2. So we're going to learn more in A2 when talk about transcription factor. So they're essential for regulation of gene expressions. So they bind to the promoter regions so that DNA adjacent to the gene can be regulated. So the first nucleotides at the 5' new mRNA chain initially retain the triphosphate group. So during the elongation stage, so each addition of free activated RNA nucleotide is incorporated at the 3' ends. Yeah? Okay, so the first one, always with the 5'. So the next one, we add the 3' end. Continue and continue. Okay, so elongation of RNA continue until the termination, where RNA polymerase recognize termination sequence. They consider specific sequence of the base of DNA template. Okay, so it signals the separation RNA polymerase from the template, and then newly synthesized RNA can be released. Okay. Now we do know that for eukaryotic cells, right now, guys, this is only in eukaryotic cell. Okay, so in eukaryotic cells, we have this after the mRNA is produced. This mRNA is called premature mRNA or precursor mRNA, or in short, we call it pre-mRNA. Okay, so in this case, what actually happened? Now look at this. This is our DNA template strand. Okay, so they have promoter and also the termination sequence. Okay, okay, the termination sequence. Okay, now what actually happened here is, if in this process, our RNA polymerase attach the promoter and start to copy down, copy everything. Okay, you copy everything until they reach this mRNA termination sequence. Then only this polymerase or RNA polymerase detach. Okay, okay, and detach. So what actually happened here is, can you see that this termination sequence won't be copied, promoter won't be copied. So this is the prematured mRNA. Are you clear? So it formed this what we call the prematured mRNA or pre-mRNA. Clear? Now this pre-mRNA consists of exon and intron. Now this is something new for you guys. Okay, something new for you guys. Why? Because you guys haven't, or didn't never learned about this before. Okay, our DNA in this case have two regions. One is called coding region. Coding region is needed, okay, for the polypeptide synthesis. So this coding region is termed as exon. Okay, termed as exon. Then we have non-coding region. This non-coding region is not needed, so it's called intron. So because it's not needed, so therefore we have to remove it. Okay, we have to remove it. So you can see that in this process, we make a band like this so that we can combine the needed one, remove unneeded one, and not needed one. Are you clear? So this is the process called intron removal or removal of the introns. Okay, and then you can see that at the 5' here, we add a cap. So this cap is called 5' cap. Okay, I will tell you guys why. Okay, later, right? 5' cap. And then at the tail here, we add this what we call poly-A tail. A stands for adenine. So they add a lot of adenine. A lot of A there. Okay. So the reason here is to add the cap and to add the tail is very, very important here to stabilize the mRNA molecule, because mRNA molecule is very, very fragile. Okay, we need to stabilize it. Why we need to stabilize it? Because later they're going to move from the nucleus, go out of the nucleus. So a lot of enzyme. So this enzyme can actually degrade the mRNA. So in order to stop or to reduce the chances or the risk here, therefore we need to put a cap. 5' cap and in this we call the poly-A tail at the 3' end. Okay. So now next, after they have done this, stabilize it already, and also with the presence of 5' caps and the poly-A tail, it can be recognized by ribosome later. Okay. So now they remove the introns, then join the exon together. So when they join the exon together, then we form the final matured mRNA. Okay, in the nucleus, then they're going to move out and then bind to the ribosome and start the translation process. Okay. Okay, so let's look at this. Okay, so bacterial mRNA are usually immediately used after transcription, because after the transcriptions, this mRNA straight away enter into the cytoplasm. So in the cytoplasm, then you can see that at the cytoplasm, straight away the 70S ribosome. So 70S ribosome are going to 70S ribosome straight away will translate this mRNA. Okay, so now without further processing, but in eukaryotes, the original transcript known as the precursor mRNA, okay, or we call it as the premature, mRNA. Okay, so Means that for eukaryotic cells, we need to modify in several ways. It is still in the nucleus. Okay, so this post-transcriptional modification and processing activity produce a mature mRNA for the transport in translations. Correct not? Okay, now why? You try imagining that if this length contains some of the item that we do not use. Let's say, for example, this tree we do not, we have to remove it. If not, then your polypeptide will be very, very long and in fact useless. Okay, we can't fall into the precise three-dimensional shapes and so on. Okay, so now what is a modification? So modification always begins with the growing RNA transcript. So RNA transcript here is actually referred to the premature mRNA. Okay, so we add about 20 to 30 nucleotide long. At that point, enzyme add 5' cap to the 5' end. So this 5' cap is special nucleotide. Is unusual special nucleotides known as 7-methyl guanosine. Okay, so it's known as 7-methyl guanosine. So which is linked to the mRNA transcript by 3 phosphate groups. So if you look at this diagram again, can you see that 3 phosphate group? They link guanine. So this guanosine, okay, adds on to this 3 phosphate group. So why we need to do this? Very important. If a capping may also protect, okay, the degradations of the mRNA by some enzyme, and also make sure that ribosome can bind to the capped mRNA. Okay. So if you look at this, if you compare between the mammalian cells, about 10 hours. But if you compare the bacterial cells, 2 minutes only. So it means that with the cap, with the capping. So with the capping here, okay, with the capping in this case, then we will be able to prolong the lifespan of the mRNA in the mammalian cell. So it's a kind of protection. So after that, we have the second modifications here, which is known as polyadenylations. Poly means many, adenine. So we add a lot of adenine there, usually about hundreds to 200 adenine nucleotide. So this long the polyadenylated, okay, is known as polyaethyl. Okay, polyaethyl. So what is the function? So in this case, just now we have to withstand the degradation, but the tail here actually helps to, in terms of exports the mRNA from the nucleus, stabilize the mRNA against degradations, okay, and also initiation of the translations by helping ribosome to recognize the mRNA. So we have 2 processes here, one at the cap, another one at the tail. Cap at the 5' end, tail at the 3' end. Now, after that, we have to know that the typical eukaryotic gene may have multiple non-coding regions known as intron, okay, as opposed to the exon, which are the part of protein coding sequence, although the number varies. When the genes that contain intron is transcribed, the entire gene is copied as a large RNA transcript. So the pre-mRNA. So a pre-mRNA molecule contains both introns and exon sequence. For the pre-mRNA to meet the functional message, it must be capped, polyaethyl added, and intron was removed, exon must splice. Splice means join, right? Together. So previously I have told you guys, right? If this is the gene, so we have region 1, region 2, region 3, region 4, region 5. So sometimes region 2 is non-coding, region 3 is non-coding. So then we only need to have 1, 4, and 5. Can you see that? So this is the final one. So this final mRNA, we have the 5' cap, then with the 3' polyaethyl. So it becomes a matured mRNA. So this matured mRNA is going to leave the nucleus. Again, and then reach the cytoplasm. Okay, so with this, I'm done for the transcription process. So now I'm going to stop the recording.